Started here. Thank you all for coming today. This will be the last of our, I actually have no idea what semester it is, spring semester seminar series, and maybe our last full-on Zoom seminar. I think our next seminar for the next semester will be in person. So we hope to see a lot of you in the teaching room with snacks. So anyway, I'm going to let Victoria, who is also a student here and a member of our seminar committee, introduce today's seminar speaker. Victoria, take it away.
Yeah, good afternoon, everyone. So today we have on board Dr. Mustafa Ojonuba. He graduated with a bachelor's degree in agriculture, and his master's degree was in crop protection from Ahmadu Bello University in Nigeria. He also graduated with a PhD in plant pathology from the University of Florida in 2019. And he came to TREC as a postdoc from 2019 to present in the plant pathology department. So he said he likes research and developmental projects to advance solutions and reduce poverty, and he dislikes everything that is bad. So I'm looking forward to this presentation, and I want to thank him for choosing to share some of his research with us today. And I don't know if he's going to tell us during his presentation, because he's leaving TREC soon, and hopefully he talks a little bit about his future plans with us as well. So probably in the future we can invite him for another talk in one of the TREC seminar series. So when you're ready, you can go ahead with your presentation.
Okay, thank you, Victoria, for the introduction. You can hear me, right, everyone? Yeah, so I'm just gonna give a brief my insights into what I've been doing here at TREC. Thank you for hosting me all this while and having to deal with me. It has been a very good stay, and I've enjoyed a good, I think all my stay here. So the mangoes and the soccer and everything has been fine. So just some insight into what I came here to do. The title of my talk today is going to be a journey in small molecule-based disease control, evolution of efficacy, and against bacterial spot of tomato, and their mechanisms of activity. So, I mean, this is from my previous slide, the first talk I gave when I came to TREC. We all know that the history of tomato has been one of something that was previously poisonous, deadly, and not acceptable by the human race up until around the 17th century, where tomato began to be consumed. And today, tomato is one of the world's leading vegetable, just after potatoes, if you count potatoes as vegetables. So in Florida, Florida is the nation's leading producer of fresh market tomatoes in the US. And production have several constraints. It could be from inputs and also from disease and insect pests. I'm going to talk mostly on a pathogen, a disease of tomato, which is bacterial spot of tomato. And on the fruits, it appears like this. You have dark brown lesions, spots on the fruits, which kind of coalesce to form large dark lesions. On the leaves, it appears as perforations, especially if it is caused by the species I'm going to describe very soon. And if you plate it on a nutrient agar medium, you're going to have yellow mucoid colonies on the growth at about 24 to 48 hours after plating. So 4 different bacterial species cause this disease: Xanthomonas vesicatoria, Vesicatoria perforans, and Gardneri. In Florida, Xanthomonas perforans seems to be the most dominant species causing bacterial spot of tomato. And it accounts for this perforating signs you see on the leaves of tomato at early stage of infection. So in Florida, apart from the fact that Xanthomonas perforans is the dominant species. Different groups also have been established for xanthomas perforans. In a previous study in 2015, Shores et al. Classified the different groups into groups 1A, 1B, and 2. I mean, there's a recent improvement, but basically these groupings are mostly out of the effector contents, have some similarities in terms of effector contents of the bacteria. Well, that's not the purpose of our talk today, but what we just are driving at is that there are multiple lineages that have been driven by recombination in Florida, and there's no durable resistance to the bacterial spot pathogen in tomato. And due to application of copper-based chemicals, copper resistance or tolerant strains have developed in the population. So it is difficult to manage in the field. So there are alternative strategies that have been looked at into managing the disease. Use of SAR, systemic acquired resistance inducers. They are promising, but they have effects on yield in that they reduce the yield in most instances. And there have been trials to see if nano formulations can help in managing the disease of already known compounds. It's still yet to be deployed commercially. Gene pyramiding kind of breakdown very soon. Then GMOs, BS5 gene, for example, is promising. And different novel chemicals and formulations have been tried against the bacterial spot disease. It's still coming up, and most of them are used at experimental level. Also, small molecules, which I'm talking about today. So what are small molecules? The central dogma of DNA is that there is, if there's transcription of DNA to RNA, then after transcription of DNA to RNA, what you have is translation of RNA to protein. So this is the natural flow of DNA to proteins, and it's called the central dogma of DNA. However, not all products of translation end up in proteins. Sometimes you have RNA translating into short peptides. And these short peptides are low molecular weight compounds, often less than 900 Daltons in weight, and they permeate intracellular spaces and are very important in the activity of an organism. So some of these peptides have been used extensively in the medical industry as antimicrobials and anti-cancer agents. And we wanted to test how we can use them against this bacterial disease of tomato. So my duties in this position mostly is to test the efficacy of selected small molecules in their ability to improve management of bacterial spore of tomato, and to also evaluate their mechanism of actions. So before I joined the lab, there was a trial done on Cavacro to see if it could manage the disease, and which was published. And also, when I came in, so I tried 4 other small molecules. So I look at eugenol, tranexamic acid, ferulic acid, and caffeic acid. If you look carefully, the molecular weights of all these compounds are below 900 Daltons. So they are effectively small molecules. So these other 3, the tranexamic acid, the ferulic acid, and the caffeic acids, belong to a group of compounds known as cinnamic acid, which I will explain later on. So 2 terms I'm going to use often in my presentation is MIC and MBC. MIC is short for minimum inhibitory concentration, which is the minimum concentration that prevents the visible growth of a microbe. So for example, if you have a chemical applied to a microbe, it is not just immediate, that's at different concentrations. Not all concentration is going to kill the bacteria, and not all concentration are going to allow for visible growth. So for example, if you have 8 microgram per mils and 4 microgram per mils and 2 microgram per mil, with all this concentration up to where you did not have any applied drug, what you find out is, in the end, at certain point, you find out that you not find visible growth of your bacterium. So for example, if you plate out at this point and you see visible growth of the bacterium, and you plate out the next concentration and you don't see visible growth of the bacterium, The point where you don't see the visible growth of the bacterium is called a minimum bactericidal concentration. So it kills the bacterium entirely. But at certain points, you wouldn't see growth of the bacterium, but if you plate it out, you're going to find that there are few bacteria inside cells inside the treatments. So at that point where you don't see visible growths and you plate it out, yet you see some growth of the bacterium, is called minimum inhibitory concentration. So I'm going to use it a lot in my talk. So I'm going to take a brief detour to talk about surfactants in plant disease management, even though my talk is mostly on small molecules. And why is this? So surfactants, when I joined the lab, I found that surfactants are used in combination with chemicals for controlling bacterial diseases. So you mix it with the chemicals. The purpose of applying surfactant is mostly to spread the chemicals so that it reduces the surface tension between the leaf surfaces, and it can penetrate the plant surface and go to the target sites. And their really intended use is not for direct control of pathogens. So there are different types. It could be synthetic surfactants, it could be biosurfactants. In the lab here, we have mostly synthetic surfactants. So we have like 4 of them or more. We have Silhouette, we have kinetic, cohere, and induce. So my intention was to see these surfactants, do they just have the spreading properties against with the chemicals, or they have other properties that have direct effects on the bacteria we are studying, So I wanted to test to see if these surfactants have some effect on them. So I went ahead and test the MIC and take the bacterium in vitro to see if they have MIC, if there's a point where you come and you won't see the growth of the bacterium, or there's a point where it kills the bacterium. So I did for Silhouette, kinetic, and cohere, induce. All these are product names. And I found out that, yeah, surfactants have effects on bacterium. So it's not only spreading the chemicals on the plants to their target sites, it has its own effects on the bacterium. So how does this effect on the bacterium, how does it happen? What is the function of this surfactant in plants? If it can kill the bacterium in vitro, so what happens in plants? So we went ahead and tested the surfactant also in plant system and tried to see if it has any advantageous effect against the bacterium in the plants. But what we found was different from what's happened in the in vitro studies. So previously, we found that all the surfactants that were tested, all of them were effective in killing the bacterium. But once you apply it to the plants, you're going to see different effects on the plants, such that in the end, you might end up increasing the pathogen, the disease, instead of trying to kill the bacterium, because the surfactant itself have things that helps that wear off the plant surfaces and reduce the surface tension, making it easy for them to enter into the system and increasing the bacteria population in the plant system. For example, if you take Silhouette, for example, Silhouette was even the most surprising of this. Silhouette, in our first trial, we found that the disease level was around 93 on our percentage scales, 93% of the leaf surfaces. In fact, in the second and third and fourth trial, we couldn't even measure. It caused burn on the plants and at the MIC level. So it was not even advisable to use it alongside the chemicals. But some other chemicals like kinetic, the best one we had was actually cohere. Cohere was gave comparable result to the control. The control, we didn't apply anything. So it was just the bacterium. So, and we found out that in most cases, the cohere treatment with cohere, which had similar results with the control where nothing was applied. But for the others, they are mixed results, kinetic and induced. In some cases, it's the same. In some cases, it's different. From this study, what we can glean out is that different surfactants have different effects on the bacterial spots in the plant system, not what we see in vitro in the laboratory. We went to the field and looked at Cohere, if it has any control of bacteria in the field system. And this is a 1-year trial, and we found that comparably with Coside, which is the standard what the growers use on the farm, it's the disease levels is similar and is different from the control, which was really a promising result for us to use in our future studies, since we intend to combine surfactants with our chemicals. So we looked at probably Cohere would be a good option for us to use in combination with our surfactant, with the small molecules in our trials. So let's get back on the road for small molecules. So initially I said we had eugenol and other chemicals which I was connected. So a brief background on eugenol. So eugenol is a phytogenic bioactive compound and is found in several plants. So in clove is the reported plant with the highest concentration so far, about 180 milligram per gram. But other plants, which actually has a lot of concentration also, we have the clover pepper and cinnamon, tulsi, turmeric, nutmeg, and thyme, all have concentrations of eugenol at different levels. So eugenol has been tested in human system, and it has been shown to be effective in managing oxidative stress and also in disrupting microbial DNA and protein, and also as in reducing elevated cholesterol levels and so on. So I went ahead to look at the MIC and the MBC of eugenol against bacterial spots, that's Xanthonas preference in vitro, and we determined it to be 1024 milligram per liter will even kill the bacterium in vitro. In previous studies, eugenol have been shown to enhance resistance in tomato against tomato yellow leaf curl viruses. So most of our studies were we also do some preliminary tests to see if it is better to apply it as an induced resistance or inducer of resistance or post-inoculation. But we stuck from our trials, we stuck to the pre-inoculation application of eugenol now at most of our trials. So this is results from greenhouse trials, trial 1, 2, 3, and many more. I just picked the ones for this talk. But what is easy to see from this table mostly is that in trial, for the most part, applying eugenol 1 day before inoculation gave best control against bacterial spots. So we had 7 days before inoculation, we have 3 days before inoculation, and we have 1 day before inoculation. The DBI is days before inoculation. So if you compare along, we see this is the control. Sorry. So in the control, we find that in trial 1, for example, we found out that applying eugenol 1 day before inoculation gave a better control of bacterial spots than the control where nothing was applied. The same thing in trial 2 and in trial 3. Even though they are similar to other treatments, but we give our ratings mostly against the control where no chemicals was applied. So we found out that we established from some of these trials that applying, first of all, eugenol provide control against bacterial spots in the greenhouse, and secondly, if you apply eugenol 1 day before inoculation, you're likely to get the best results against bacterial spots. So we also tested if we can combine this with surfactants and the eugenol surfactant to see if we're going to have improved disease management against bacterial spot. So we have both. So we're thinking that if the surfactant is going to spread the eugenol, in addition to providing control against bacterial spots, but it is not always the case. So we found out that in fact, it is better to apply eugenol without the surfactant. You end up getting better control at the MIC level. If you're going to apply the surfactants at levels below MIC, probably you just get a better spread. But we were just trying to see if we were able to get control at MIC level, both with the surfactant and also the eugenol. So anyway, so if you look at this place here, these are the surfactants, kinetic and cohere, and these are the eugenol treatment without any surfactants, and these are the eugenol treatment with different surfactants. What we found was that applying eugenol without surfactants gave you a better disease reduction than the control here when you applied nothing, and also compared to other treatments where you have both eugenol and the surfactants in mixture. So in both trials where we have here, it was better not to mix eugenol with surfactants at their MIC levels. Oh, okay, sorry, I missed this point. So that will give us a background for the greenhouse evaluation of eugenol. And I went ahead and also did experiments with cinnamic acid. And just a brief background about cinnamic acids. They are a group of aromatic carboxylic acids that apply. They are very, very prevalent in many plants and even microbes. And the name cinnamic acid actually comes from cinnamon. They are formed in a pathway that have a lot of chemicals that are produced in the process. This is a snapshot of one of the pathway in plants. So here you see that this phenylalanine pathway is also called the cinnamic acid pathway. So the enzyme PAL, which is phenylalanine ammonia lyase, introduce a double bond into the phenylalanine and form cinnamic acid. From this point, a series of either methylation or adenine hydroxy groups form different kind of compounds that have found use in both human medicine and also in plant management systems. So we have fumaric acid, you have ferulic acid, hydroxyferulic acid, you have different kinds of chemicals that are produced in this system that have been used and tested in plants in human system against bacteria, against known diseases. So for us, we are focusing on three of these. We focused on tranexamic acid, caffeic acid, and the ferulic acid. And as usual, the first thing I did was to determine the MIC and MPC for these cinnamic acids. And what was the highlight is that there was the MIC was unusually high. We have like 8 grams per liter for the MICs and 4 grams per liter for the MICs of ferulic acid and caffeic acid. I mean, when I checked the literature, I found that it is not unusual for high MICs for cinnamic acids. Well, I went ahead and conducted greenhouse trials also to see if we're going to have disease control against bacterial spot with the small molecules, the cinnamic acid, ferulic acid, and caffeic acid. In the first trial, we had it's shown here that for the most part, the tranexamic acid was the most effective, and also the caffeic acid provided a good level of control against the disease. But the ferulic acid, even though it gave a lower disease rating, was not very much different from the control. The same thing in the second trial. The role for the values for caffeic acid and ferulic acid were reversed. The caffeic acid was lower than the control, but it was not statistically significant. Ferulic acid in this case was significant, but tranexamic acid was constant and provided control against bacterial spot in the greenhouse. Well, from this place, we can really see that to a very large extent, eugenol, tranexamic acid, ferulic acid, and caffeic acids gave good control against this bacterial spot in the under greenhouse condition. So the next thing was to try this, what we've seen in the greenhouse. We'll check the field if we can use it and get the same results, and also possibly have it as part of the tools to recommend in the future. So we had field trials. I'm just going to present the results from, we had 3 of them. I'm going to present 2 results from the field trials. What is clear from this table is that the eugenol, and we have different treatment. We have eugenol with cohere, eugenol with coside, ferulic acid, and caffeic acid. All the treatments for eugenol, the ferulic acid, the caffeic acid, we gave better, lower disease rating than the untreated control. And even if you look at the AUDPC, you find the same thing. They were significantly very different from the disease, the untreated control, showing that, yeah, the eugenol and the other, the ferulic acid and caffeic acid provided good control against bacterial spot. And there was not much effect on the yield. As I said earlier, some chemicals have been tested before and they have effect on the yield. But in this case, the yield probably they remain the same for the most part. So there was no much difference in the yield. But we had a little bit a better control against the bacterial spots than when you didn't than the untreated control. The same thing we found in the second year field trial. We had the same similar results where ferulic acid, caffeic acid, and the eugenol provided better control against bacterial spots significantly than the grower standard, which is a mancocide, coside or mancocide, or the untreated control. Similarly, on the AUDPC, which is the area under disease progress curve, we have the same similar story to tell. But again, the yields were not so much different. Probably the plant variety can withstand bacterial spot onslaughts. So from this, we conclude that eugenol, tranexamic acid, ferulic acid, and caffeic acid gave significant disease controls under field condition. So my next target was to see how does this chemicals function. What is what mechanism do they deploy in attacking or helping to manage disease, is whether in vitro or in the field. And so it's a large study. I'm not going to spend much time on this place because there's lots of data, a lot of them still under processing. But I'll talk a little bit on the metabolomics aspect, and hopefully we have an insight into how this one of these chemical function. So I'll talk mostly on Cavacrol because it was done. I mean, we have published data on that, and we want to see how does Cavacrol kill the bacteria? What are the systems that Cavacrol interact with when it wants to kill the bacteria? So we had a time series studies where we had the reference strain of Xanthomas perforans, which was XP91118. We had the reference strain and we treated it with Cavacrol. We have an untreated control, and we took samples at 1 hour and at 6 hours. And for each of the treatments, we have 4 replicates. And we also have a 5th replicate, which was drawn by pulling the 4 samples together. So these samples were prepared. I mean, it's a long process in terms of preparing materials. And we use the LC-MS, liquid chromatography mass spectroscopy, to analyze our data, to run it on our data. So mostly it runs both in positive and negative ionization phases. And the good thing about using this process is that it gives you a kind of view of the totality of the metabolites in the system. and help you to discriminate those one which are upregulated and those one which were not highly regulated in a particular pathway as you applied the chemical or as the chemical acts upon the bacterial system. Well, when we got our result back from the metabolomic score at Gainesville, we saw that there's a huge amount of metabolites in the xanthomonas perforans system. In fact, in the positive phase, about 1,477 metabolites were identified, and in the negative phase, about 1,094 metabolites were identified. Not all of them were known. We just, from what was gotten, we only knew 125 in the positive phase and only 109 in the negative phase. So I had to go through the databases on the human metabolome database and the Methylene. These are two databases that have a lot of metabolites that have been curated in them, and they are procedure for searching against these databases to see if what you have is also present in them and identifying what you have. It's a process of annotation that requires searching for the mass of the metabolites you have in your data sets. Anyway, so among the significant metabolites that we got, we knew 67, and in the positive phase and negative phase, we knew 55, right from when it was sent to us from the metabolomic score in Gainesville. And you don't have to worry yourself with this. This is a list of some of the metabolites that we got that were significant in the positive phase, and these were some of the list of the metabolites we got in the negative phase, where that are significant as a result of treatment with the cover crop. So what we found out was really interesting, because the first work that has been done on xanthomonas perforans in terms of metabolomic studies, and we found a lot of chemicals that for the first time being reported in microbial systems, and some chemicals are for the first time being reported in xanthomonas systems. So, for example, we have cementosin. Cementosin is has been always, in all the literature that we searched, it's only present in plants and butterflies. This is the first time we are seeing it in a microbial system. And also, for example, if you look at methoxybrasinin, for example, methoxybrasinin has never been reported in any microbial system. It's in brassicas, and we are reporting it for the first time in a microbe. And for all this one, Haman, beta-carbolin, and piperidine, and maculocin, pyrocol, these are the first time we are reporting it in the xanthomonas system. So most of these studies are still ongoing, and it gave us insight into the amount of metabolites that are present in a bacterial system that people have probably ignored for a long time. In fact, there are some metabolites that have been gotten from different systems that have been well known in the pharmaceutical industries, like crutameton, valsartan, methylbutal. These are metabolites that have been turned into drugs in the pharmaceutical industries, and we found them that this small bacteria, not small, but this bacterium that we look at as a plant pathogen has these enormous chemicals in it. So it's a lot. I'm just giving a snapshot of what we found, and it's really, really mind-boggling how a bacterium would have these sets of chemicals in them that could be explored for other benefits. So, and it is not that what we found, we can really, it's really at high level of confidence, because the chromatograms were really definite that there were these chemicals. So there was, it's a matter of trying to analyze more and see what type of, or what form are they appearing, but we are sure that these chemicals are within the system of xanthomonas perforans. So before I continue, so some of the things we discovered, the chemicals we discovered, the piperidine, the pyrrolidine, we have an MS student, Katsira, who has tested some of these chemicals, or who is testing. This project is feeding into her project, and she has shown, I mean, very confidently that these chemicals that we got from Xanthomonas preferens can even be used to control the bacterium. So piperidine, for example, and pyrrolidine, for example, 1 hour after you apply them, the bacteria, I mean, dies. I mean, this is compared to the copper, for example. You see still some bacterial growth, but for pyrrolidine and piperidine, just 1 hour after applying them, if you plate them out, if you plate the solution out, you don't see any bacterial growth. So she's working on it for her PhD, for her MS, and I'll leave her to talk about it when she graduates. So back to the data I got. So in the positive ion mode and the negative ion mode, we found that, yeah, there's a lot of differences in metabolites that at 6 hours, 1 hour, and the control that we didn't apply any Cavacro, and the PCA really described the differences which we see in our analysis. Well, what we did was to overlay the significant metabolites that we got in the positive ion phase on the bacterial, the Xanthomonas preferens metabolic pathway. So it's known, it's a sequence genome. So we have it on NCBI, also have it on BioSeq. So we got with this, what they called metabolic pathway analysis. We overlay these chemicals, the metabolites we found, over the pathways that are present in Xanthomonas preferens. And what we found, very interestingly, is that these metabolites, the Cavacro probably have effect on a large number of pathways. It's not going to be one pathway. So we see that amino acid biosynthesis, it's affecting it. We see that here, amine and polyamine degradation is affecting it. Amine and polyamine biosynthesis is affecting it. So there's a lot of pathways that the Cavacro must have interacted with in its activity in killing the bacterium. So the same thing we see in the negative phase. The TCA cycle, for example, here is affected. The nucleoside and nucleotide pathways are also affected. So the same story, a lot of the Cavacro affected a lot of pathways in Xanthomonas preferens in its activity against the pathogen. So what we did, we used a platform called Metabolomics.ca, which we used to analyze some of the significant metabolites to see if we can come at a point where we can pinpoint some pathways that are probably we can hold onto as guilty, kind of that are being involved in the activity of Cavacro. Well, we couldn't find Xanthomonas preferens in the database, but we used a different microbe, Pseudomonas bifida, which has some common pathways with Xanthomonas preferens. And when we did the analysis using Pseudomonas bifida, we found out that in the positive phase, at least 14 pathways were being affected strongly. So when I mean strongly here, I labeled 1 to 14, but I didn't label these ones. 1 to 14 have at least 2 metabolites in the pathway that was affecting the pathway. So we, for example, the first one, arginine and proline metabolism, probably have like 7 different metabolites, but we choose the one that have at least two metabolites acting in the pathway. So we found out that at least 14 different pathways were being affected very strongly by Cavacro. In the negative ion phase, it's similar story, even though there's a mix of the pathways. But what's the find that there's some common pathways and also some different pathways. So, but in all, if you put everything together, we can say that at least Cavacro acts on at least 18 different pathways. Whether it is biosynthesis, whether it's degradation, but based on what we found, at least 18 different pathways have been affected on. But 7 pathways are common in both the positive and negative ion phases: pyrimidine metabolism, amino acyltRNA synthesis, sulfur metabolism, pantothenate and CoA biosynthesis, and the rest. So we are pretty confident that at least we have a common ground to say, I mean, a very confident result to see that at least 7 different pathways have been affected by Cavacro in its activity against Xanthomonas preferens. And the highlight from this mostly is, if you look at both the positive and negative ion phase, pyrimidine pathway is commonly affected in both pathways, and also in the negative phase you have the purine pathway. So these are DNA pathways, pyrimidine and purine, and they make up the DNA. So it means probably that Cavacro is involved in either DNA damage, maybe, or maybe it's involved in DNA protection. I mean, maybe there's a mechanism set up by the bacterium to protect the DNA, and that is why we have a high or elevated number of pyrimidines and metabolites in the pyrimidine and purine pathways. Probably that's the reason. We don't know. Well, we went ahead to look at the to see how this varies. I mean, we did some analysis to heat map to see if we find a direction to how we can see if these metabolites, how they vary at zero of the control without treatment, 1 hour after treatment, and 6 hours after treatment. So this is a snapshot. There could be a lot more graphs from that. But I want us to focus a little bit on the red arrows, for example. If you look at the red arrows here, you have uridine, you have uracil, you have adenosine 5-monophosphate, you have uridine 5-monophosphate, you have guanosine 5-monophosphate. And the same thing here, uridine, the same thing in the negative ion mode. These are nucleotides and nucleosides, so they are DNA components. And so for the most part, at 6 hours after treatment, there's an elevated level. So here the intensity is 1, shows high level, and -1, the blue, is a low level or depletion of the metabolites. So at 6 hours after treatment, there is an accumulation or elevated level of this DNA, the nucleoside and nucleotides that are supposed to be involved in DNA pathways, except for cytidine, which is at low level. The rest of them are at high level after 6 hours. And what we know is that from studies that have been done before, once you have accumulation of nucleotide or nucleosides, as it's mostly as a result of defense mechanism by the bacterium against DNA damage in other systems that have been studied previously. So probably the Cavacro does not directly affect the bacterial system, the nucleoside and nucleotide system. Probably it triggers an effect where there's an elevated level of nucleotide and nucleoside, and this helps in protecting, trying to protect the bacteria from dying as a survival strategy. We don't really know. But what is very clear is, if you look at the green arrows point to pyridoxine, sugar alcohol, the keto hexose sugars, the fructose here, and also some carbohydrates here, and some disaccharides here, and the aldopentose here. So for the most part, if you look at the glucose and the hexose, you look at pyridoxine, the sugar alcohol, for the most part, at 6 hours, they have reduced intensity. So they are blue. So they're, except for 2 of them, which are high, for the most part, they are blue. So it means that Cavacro actually also affects the energy synthesis pathway, because the energy is being used up at 6 hours after treatment with Cavacro. So it's very low in sugar. It's very low in the keto hexoses. It's also very low in glucose. Even at 1 hour, we can already see the effects here, for example. So it's it appears that carbapenem is limiting the biosynthesis of energy in xanthomonas preference. So from this is a summary we can glean from this study. I mean, there could be other ones. We could look at the effects on other ones, but at least this gives us an insight into how probably what carbapenem is doing against the bacterium that is killing it. So from this, we can conclude that accumulation of nucleotides is related to DNA protection. Probably this carbapenem, there are some things that the depletion of energy metabolites is making the bacteria to look for regions where it can, or to derive things to increase by accumulating a lot of nucleotides to protect itself against what is happening in other systems. Well, we are doing other further studies using RNA-seq and qPCR, and I wouldn't. So those are also in the pipeline. So, but this is a snapshot of what we got from the pathways that's a possible effect of mechanisms of carbapenem against bacterial spot pathogens and Xanthomonas preference. So with this, I'd like to thank everyone, my lab, especially my PI, Dr. Zhang has been wonderful, and Dr. Liu with a few trials. Yuan Wang helped me a lot in planting the planting materials and preparations in the greenhouse. Katsira Yi, Dr. Fang, who is currently in Taiwan. We all work together. My collaborators, Dr. Jeffrey Jones, Gerald Winsavage, Nush Sharma, and the guys at the Metabolomics core in UF, Timothy Garrett, Joy, and Dr. Mahmoud Ahmed, and Laura. Thank you, everyone, and thank you for listening.
Thank you so much. That was a very interesting topic. Yeah, this is actually an eye-opener for me, especially in the aspect of my research. Like, I'm actually seeing the possibility of probably combining, using some surfactants with some of the insecticide I use to see probably if there's an additional effect or, synergism. Because from your result, I see that it does not always have a positive effect, I hope I'm right about that. Yeah, I'm sure like there's a lot of questions, and I would like to give room for questions if there are any questions. You could send your questions in the chat, or you may go ahead, unmute yourself and ask your question. Are there any questions?
Or I can go ahead and present again.
Are people still online?
Yeah. So I was so very clear, I should say.
Are there any questions from anyone?
Mustafa, I'll ask a question. So this was really interesting and very outside of my wheelhouse. So it was a lot to take in. But so I'm wondering if, like, my lab, we study coastal ecology and nutrient cycling, and I wonder how you might be able to see any synergies between the work you've been doing here and what our lab is doing.
Yeah, great. I talked with Bella some times ago when she was talking about her project, and it's something that is there's a lot of synergy in all these things, because now I think, if I'm correct, looking at coastal, I mean, she was looking at coastal stuff. I've forgotten the details now, but yeah, one thing I could also see from that is to see the different coastal, areas you have, for example, I would think that they have different populations in terms of microbial bodies, and they would have different effects, probably maybe the different treatments or that is happening, or the way that the different coastal systems have different effects on the dynamics on the microbe you find in the system. And eventually, this will, result in a series of interesting topics where you want to see if the microbes in the system have responses to different temperature levels in the coastal areas, different how will I put it now? Whatever effects, whatever physical effects that happens in those areas you're studying could have different effects on also the life forms in those systems, where you want to see if, for example, activities that happen during the hurricane seasons, for example, have any differences on how the life forms in those coastal areas survive compared to when there's no hurricanes or other things like that. So I don't know. I've forgotten. I think I used to know the details of what Bella was doing earlier, and I could see something that could come out of it. But I think since I left the office, I have not been interacting with her that much. But yeah, I think since it involves a lot of studies in terms of probably the life forms in those coastal areas or whatever it is, I think there's room for application of RNA-seq, for metabolomics, and for other things to see if you can discover a lot of different happenings within the system, especially gene regulation.
Great. Bella, I hope you're taking notes. We might follow up with you.
Yeah, there's a question in the chat. The question says, for work with Cavacrow, what suggestions do you have for any work in the future if you continue in this aspect?
Oh, yeah. Cavacrow is, Kang and Dr. Liu did a lot of work on Cavacrow in terms of the efficacy in the field and also in the greenhouse. But there's a lot more that I think can be done, especially collaborating with those in the human medicine field, because some of the things we found is Cavacrow is known to be an anti-cancer agent. It has been tested for anti-cancers, for example. But the chemicals we are finding from the system, bacterial system, probably could have some potent effect also in against some human diseases, which we don't know. So I would really think that Cavacrow the formulation needs to be, for plant system especially, there have to be some effect on formulation, what's reformulated, and also long-term to defeat the bacteria, long-term to see what concentration could induce resistance in bacterium. So, I didn't do that, but probably do we know if you are spraying Cavacrow, for example, the bacteria does not get the equal spray all the time. So some of them are going to escape because we didn't kill all the bacterium in the plant system. So definitely some of them got the Cavacrow but still survive. So what can be done to mitigate against future resistant development is another field that can be looked at. So formulation, especially if work is supposed to be, if commercialization is being thought of, then formulation is really key to see what can be deployed in the field for farmers' utilization, growers' utilization.
Thank you. A quick question. Do you know what the breakdown products of Cavacrow are and whether this may have similar effects on your non-target organisms?
I don't know. Yeah, that's a good question. Thank you.
Yeah, and the last question, because our time is almost up. Are surfactants same with adjuvants? They are not, right? Because adjuvants sometimes comes with pesticides and sometimes does not. So are surfactants same with adjuvants?
Yeah, there are words that can be used interchangeably. In most cases, some use adjuvants and some use surfactants. But in some cases you have pre-formulated. Once you buy your chemicals, you have them already formulated with the surfactant, and in most cases it stands for the same thing, but there could be slight differences.
Thank you. I want to believe there are no more questions, and we really want to thank you for the talk today, and we hope to see you in future seminars wherever you may. I think you didn't mention that you'll be leaving TREC next sometime soon. So hopefully in the future we'll invite you again to speak in one of the TREC seminars.
Okay, thank you. I enjoyed my stay in TREC. Thanks to everyone. And to the committees I belonged, and everyone has been really nice and friendly and helpful and collaborating. And I wish I could stay longer, but I have to move on. So, yeah, thank you so much for the opportunity.
Yeah, thank you so much.
Yeah,
and thank you all for joining today. So like Dr. Ashley mentioned earlier, that this is the last of the spring seminars, and we hope to see you in the fall series. The next one is coming up by August, and just look out for the emails and flyers for the fall seminar. Have a good afternoon.